will be having 4 lectures. I mean 4 basic topics to study, 4 basic umbrella topics to study. Within that umbrella topic you will be having many subtopics. One is biotechnology, biology and biotechnology. Second one is IT and communication technology, information technology and communication technology. Then third one is space technology, everything related to space, ISRO and India's interplanetary missions, basics of universe kind of thing. Then fourth one is related to defense technology. Okay, everything about our defense establishments, our new developments related to defense, like new vehicles for defense, new weapons, new helicopters, like submarines or missiles kind of thing. So we'll see in these umbrella topics, I mean in these modules we'll be having many subtopics. We'll see each one. So without much delay, we'll start our first lecture on biotechnology. But before that, before moving to biotechnology, we need to have a little bit knowledge about biology. So first thing everyone know is that the reason why we are having some similarity in traits with our parents is genes, right? We are inheriting genes. I mean, our grandparents contributed some of the genes to our parents, and from our parents we are inheriting some genes. I mean, not some genes. I'll tell you how much quantum of genes are being inherited from parents to offspring, and from us to our children will be giving many traits. That's why people whose parents are having like parents are taller, their offspring will be taller. Some people will be having some kind of greenish or bluish eyes, because even if their parents are not having bluish eyes, maybe their grandparents or great grandparents will be having that trait. Okay, so that kind of trait will be inherited from grandparents to parents, parents to us to our offsprings kind of thing. So these genes are made up of an organic substance called DNA. We all have heard about DNA. So we'll see what is a DNA, what is a gene, what is a chromosome, okay, what is meant by genome, where are these things located. I just want you to know these things in a very simple language. Whenever you observe the textbooks or newspapers or Google and all, these things are written in a very complicated language. Okay, I just want you to understand this thing like eating biscuit, that much simple. Okay, we'll start. First of all, what is biotechnology? Very simple. Biotechnology means you are integrating technology to biology. That means without the use of technology, you cannot see the genome or DNA which is present in our cells. For that, you need to have many laboratory apparatus. Okay, you need to have specific microscopic tools to observe these things. And with the help of technology, you will be able to edit your genome. Okay, because technology has developed to this scale, we can modify the children. I mean, normally a parent who is planning to have a baby and they feel that they are having a like very low profile capabilities. Okay, they are not very intelligent, they are not having any like big capabilities, like they are not able to create music, they are not able to act, but they feel that their children need to have these kind of capabilities. He or she should bat like Sachin Tendulkar, should be able to create music like A.R. Rahman, okay, and should be beautiful like Mammootty or Aishwarya Rai. Okay, so these kind of traits, every parents will be having that kind of a wish, so that their parents, I mean their offsprings are becoming something unique. So with the help of technology, we are able to see the gene or the genetic sequences of Sachin Tendulkar. Okay, if you are getting one cell from Sachin Tendulkar, maybe like you guys have observed using like during the time of our COVID-19 and all, they will be taking some cells from our cheeks and all, okay, or from our like nose and all, through some swabs and all. Okay, so with that you are getting cells while they are taking some samples from our cheek, from our nose, etc. They are actually collecting our cells. In the laboratory, they can open up the cell, they can open up the DNA, and I'll show you how a DNA looks like. DNA is made up of some chemicals. These are made up of 4 chemicals. I'll show you that: adenine, thymine, guanine and cytosine. Okay. every single organism's body is made up of these 4 chemicals, but its order is different. Okay, adenine, thymine, cytosine, guanine. This is one order. Adenine, guanine, cytosine, thymine. This is another order, and such in that manner we are having 3 billion arrangement of this thing. Okay, in chain these are arranged like A, T, C, G, C, T, A, T, like that it is going. Okay, so if there is one change in, if there is a small change in the order of these particular like chain, in place of A, in place of adenine, if I am having adenine in one gene and you are having thymine, you will be having more height than me. Okay, this is just introduction I am telling. I will show you each of the things in detail using pictures and images. You will understand everything. But what I am telling is, through the help of technology, you are able to change the position of these chemicals. In place of adenine, you can make cytosine, or like that kind of changes you can make. With that change, a person's trait will be different. Okay, I'll show you. This was just an introduction. So what I am telling is that these genes are present in our body, that is biology. Now using technology, you are making some modifications in these genes. This is known as biotechnology. Biology integrated with technology. So this is about thing. Now in the this is the same introduction. Like if you are taking the case of living beings, not only humans but the entire living beings, the genetic material is only 2. One, either it is DNA or it is RNA. Towards the like course of this module, we will see what is the difference between a DNA and RNA. But humans are having only one single genetic material, that is DNA. Now, where is this thing located? Where is this DNA located? DNA is present in within the cell where nucleus. Okay, this is one cell. If I am taking, this is a person, I am taking one cell from a person, like maybe a blood sample, maybe his skin cell. In all cells of the human body, whether you are taking from my hair, from my nail, from my eyes, from my cheek, from my skin, from my leg, the DNA is same. DNA is same in all the cells in one particular human being. But in the different human being, there will be some small changes. Okay, in the arrangement of these base pairs, these bridges, in the arrangement of these bridges means what? This one, adenine, cytosine, guanine and thymine. This is the order, this is the genetic code, this is the arrangement of these chemicals within a particular human being. And there will be some small difference between you and me. Only that. But the composition is same. All human body is made up of these 4 chemicals, adenine, thymine, cytosine and guanine. And we are going to see that in detail. First of all, I am taking one particular cell from a human being. You can see that in that cell we have different components. We have mitochondrion, we have ribosome. These are having different functions, and the main component or the brain of a cell is known as nucleus. Within that nucleus, you have a thread-like structure called DNA. Actually, it is seen in this form, thread form, but if it is arranged in this form, like it will be like in a haphazard manner, it will be like this. When we draw with some pen, if you can draw like this, it will be like that. But our body arranges this thread in a proper order, in a proper fashion, so that it will not be tying each other into a shape called chromosome. I am just curling. Like sewing machine thread കണ്ടിട്ടില്ലേ അതിങ്ങനെയല്ലേ ഉണ്ടാവുക ചുറ്റി like ആണെങ്കിൽ നമ്മൾ thread വെക്കുക എന്ന് പറയുന്ന പോലെ our body, our nucleus curls this particular DNA into a proper shape like this. This is the shape in which you can see the DNA within the nucleus. Actually, we are having 23 pairs of these structures called chromosome. This is nothing, this is the same thing is DNA only, but DNA is arranged into a proper shape with 4 limbs. This is known as chromosome. So chromosome and DNA are one and the same. You got that point, right? Chromosomes are DNA are one and the same, but DNA is arranged into a proper shape called chromosome. This shape in which DNA is curled and arranged is known as chromosome. Within this chromosome, you can see different genes. Okay, this may be one gene for eye color. This may be another gene for height. There may be another gene for our hairs, some people are more hairy, some people are less hairy, some people are having light skin, some people are having dark skin. So these kind of informations which are expressed in our body in the form of different traits. Maybe some people are having sharp eyesight, some people are having sharp hearing ability, some people can perceive, smell more beautifully than others. So these kind of traits are expressed with the help of the segments of DNA called genes. I'll show you what is that. See, this is the cell. This is a cell that I have taken maybe from my cheek. And if I am zooming in, if I am zooming into the cell, you can see this nucleus. Nucleus is present within the cell, and we can see that DNA is arranged in this shape called chromosome. And again, I am zooming into one particular chromosome. You can see different bands present in this chromosome. These bands are known as genes. So genes are nothing but a segment of DNA which is giving a meaningful information. Okay, if you take the case of English alphabets, we have 26 letters, right? But if I am telling A, B, C, D, E, F, G like that, there is no meaning. But if I am arranging that with a spelling called N, A, Z, E, E, B, that's my name. So you can identify me if you are arranging this in a proper order. Similarly, our body, within these chromosome bands or within these segments of DNA called genes, body will be, the nucleus will be instructing the human body with some traits. For example, if you want to have, I'll show you one thing, one second. Yeah, consider this is the order. If I am opening one cell, from eye, if I am taking one cell from the eye, maybe from your ear, eye drops, you can get one cell from the eye. Okay, when I am opening that particular cell in the laboratory setup, I am seeing that my eye cell is having in one particular chromosome. If I am opening, I am having this order. Okay, thymine, adenine, thymine, guanine, thymine, like that. When I have observed one of yours eye cell, I have seen that that particular chromosome is having a segment of gene. It is showing this order: thymine, adenine, guanine, and thymine. In place of adenine, like I had this adenine, you had this guanine. So my eye was brown, your eye was blue. So a single letter change, a single change in the order of one chemical made a big difference. This is how it happens. So this is the segment of DNA. This I'm taking one particular piece of a DNA or a particular chromosome. You can see that this much segment, it may be having this order: A, T, C, G, A, T, C, G, like that. Okay, so this is known as gene. So gene is a segment of DNA or chromosome. DNA and chromosome are one and the same. Okay, chromosome is just a shape. Okay, so one particular segment of DNA which is conveying a meaningful information for one particular character of the person. Got the point, right? This is the gene. Now, so it is having actually like this, but we are drawing it into a regular shape like this. In nature, it will be like this, curling, like in a curled fashion. So if I am uncoiling it, this was the regular shape. I am slowly uncoiling it. You can see it is like this. It is the kettupaadu koodadiരിക്കാൻ വേണ്ടിയിട്ട് ഒരു set of proteins നെ ചുറ്റും ഇത് കെട്ടി വെച്ചിരിക്കുകയാണ്. Okay, DNA is curled around a kind of protein called histone proteins. നമ്മൾ മത്സ്യത്തിന് ഫിഷിങ്ങിനൊക്കെ ഇവർ വലയിടുമ്പോൾ അതിനകത്ത് കെട്ടി വെച്ചിട്ടുണ്ട് കാണാമല്ലോ, വല കെട്ടുകൂടാതിരിക്കാൻ വേണ്ടി. സോ ഇൻ നേച്ചർ ഓൾസോ ഇതിങ്ങനെ കെട്ടുകൂടാതിരിക്കാൻ വേണ്ടിയിട്ട് DNA is curled or wound around a set of proteins called histone proteins. Maybe നിങ്ങൾക്ക് ഒരു MCQ ൽ മേബി ചോദിച്ചേക്കാം, അവർ അത്ര ഡീപ്പിലൊന്നും ചോദിക്കുന്നില്ല. ഈവൻ this is like a deep information. They won't be asking you even at this much depth. Okay, very basic thing അവർ ചോദിക്കുള്ളൂ. Very surface level അവർ ചോദിക്കുള്ളൂ. പക്ഷേ just for the extra information you can know this. Histone proteins ചുറ്റും DNA are wound around, and ഇത് നമ്മൾ കുറച്ചുകൂടി നമ്മൾ ലൂസ് ചെയ്താൽ നമുക്ക് കാണാൻ പറ്റും. Again I am opening, again I am uncurling this thing. You can see that these kind of bridges are made. This greenish, this dark green thing will be adenine, the other will be cytosine, then other will be thymine, then other will be guanine. ഇതിന്റെ പേഴ്സ് ആയിരിക്കും ഇങ്ങനെ പോയിട്ടുണ്ടാവുക. Okay, similarly we are having 3 billion such pairs. Base pairing എന്ന് പറയുന്നുണ്ടെങ്കിൽ ഈ അഡിനിൻ ഗ്വാനിനൊക്കെ നമ്മൾ കെമിക്കലി പറയുന്നത് ദീസ് ആർ ന്യൂക്ലിയോടൈഡ് ബേസസ്. ന്യൂക്ലിയോടൈഡ് ആ പേര് അങ്ങനെ തന്നെ ഓർത്താൽ മതി. ഓക്കെ, അഡിനിൻ ഗ്വാനിൻ തൈമിൻ ആൻഡ് സൈറ്റോസിൻ ആർ ന്യൂക്ലിയോടൈഡ് ബേസസ് ഓർ കെമിക്കൽ ബേസസ്. നിങ്ങൾക്ക് സിമ്പിൾ ആയിട്ട് പറയാം കെമിക്കൽ ബേസസ്. നമ്മളിപ്പോൾ കാർബൺ നൈട്രജൻ എന്നൊക്കെ പറയുന്ന പോലെ അല്ലേ, അന്ന് പറയുന്ന പോലെ കാർബണും ഹൈഡ്രജനും നൈട്രജനും ഒക്കെ കൊണ്ടുണ്ടാക്കിയിട്ടുള്ള കെമിക്കലാണ് ഈ അഡിനിൻ ഗ്വാനിൻ എന്നൊക്കെ പറയുന്നത്. അപ്പോൾ these chemical bases are forming in pairs. This is adenine, this is thymine, this is guanine, this is cytosine, like that ATCG pairs ആയിട്ട് ഇങ്ങനെ പോയിരിക്കുകയാണ്. ഇത് ഒരു പെയർ, ഇത് രണ്ട് പെയർ. ഐ മീൻ ഇത് ഒരു പെയർ എന്ന് പറഞ്ഞാൽ എന്താണ്, രണ്ട് കെമിക്കൽ ഉള്ളതുകൊണ്ടാണ് ഒരു പെയർ എന്ന് പറയുന്നത് അല്ലേ. സോ ദിസ് ഈസ് വൺ പെയർ. ഇത് രണ്ടും ചേർന്നിട്ടല്ല ഒരു പെയർ ആവുന്നത്. ഒരു ബ്രിഡ്ജ് തന്നെ ഒരു പെയറാണ് അല്ലേ, അതിൽ രണ്ട് കെമിക്കൽ ഉണ്ട്. സോ ദിസ് ഈസ് 1, ദിസ് ഈസ് 2, ദിസ് ഈസ് 3. സിമിലർലി വി ആർ ഹാവിംഗ് 3 billion base pairs of ATCG bonds. Okay, this order, any change in even one pair in place of A, if you are having T, other person is having C, the next person is having G, even that small changes makes big difference in the body. So idu orthokka. Same thing I have given one additional slide just for extra clarification. You can see that this orange represents cytosine, this light orange that represents guanine, then adenine, thymine. ഈ രീതിയിലാണ് നമ്മളുടെ ലൈക്ക് ന്യൂക്ലിയസിനുള്ളിൽ DNA കേൾ ചെയ്ത് ക്രോമസോമിൻ്റെ ഷേപ്പിൽ അറേഞ്ച് ചെയ്തിട്ടുള്ളത്. അതിലൊരു സെഗ്മെൻറ്റ് നമ്മൾ എടുത്താൽ ദാറ്റ് ഗീവ്സ് എ പർട്ടിക്കുലർ മീനിങ്, മേ ബി ഫോർ ഐ കളർ, മേ ബി ഫോർ ഹൈറ്റ്, മേ ബി ഫോർ ഹെയർസ് ആൻഡ് ഓൾ. സോ ദിസ് ഈസ് അബൌട്ട് DNA. ഇതേ ഞാൻ പറഞ്ഞ എല്ലാ കാര്യങ്ങളുമാണ് ഈ നോട്ടിലുള്ളത്. സോ യു വോണ്ട് ബി ഫീലിംഗ് എനി കൺഫ്യൂഷൻ വൈൽ യു ആർ റീഡിംഗ്. ഓക്കെ, ഈവൻ ഇഫ് യു ആർ ഹാവിംഗ് എനി കൺഫ്യൂഷൻ ഈവൻ ഇൻ ദ ക്ലാസ് ഓർ while you are reading, you can come to me anytime. I am ready to explain you 1000 times. No issues. But ഇത് ഇത്രയും പഠിക്കാനുള്ളൂ. ഇത്രയും പറഞ്ഞത് നിങ്ങൾക്ക് മനസ്സിലായില്ലേ? യു ഗോട്ട് ദിസ് പോയിന്റ് റൈറ്റ്? ഇത്രേ ഉള്ളൂ സയൻസ് ആൻഡ് ടെക്നോളജിയിൽ ബയോടെക്നോളജി. ഇനി ഇത് വെച്ചിട്ട് നമ്മൾ കുറേ കാര്യങ്ങൾ പറയും. ഓക്കെ, ഇതുണ്ടെങ്കിൽ നിങ്ങൾക്ക് യു ക്യാൻ സോൾവ് ഓൾമോസ്റ്റ് ഓൾ ക്വസ്റ്റ്യൻസ്. ഇത്രയും നോളജ്, ദാറ്റ് മച്ച് സിമ്പിൾ എ സബ്ജക്ട് ഈസ് ദിസ്. സോ വിൽ കണ്ടിന്യൂ അവർ ഡിസ്കഷൻ. Now final thing, genome. Genome means the entire set of genetic information that is present in our cell. You are not having one chromosome, you are not having two chromosomes, you are having 23 pairs of chromosomes. That means in one particular cell you can see 46 such chromosomes. 23 came from your father, 23 came from your mother. Okay, and all chromosomes are made up of genes or DNA, right? So 23 sets of chromosomes are giving you the traits from father. 23 sets of chromosomes are, 23 chromosomes are coming from your mother. Now when the trait of father and when the trait of mother combines, there will be some slight differences in your body. That's why you are not looking exactly the carbon copy of your father or mother. When two genes are combining, നമ്മൾ രണ്ട് കെമിക്കൽസ് ചേർന്ന് ഇപ്പോൾ when you are mixing some red color chemical with some brown color chemical, അത് ഉണ്ടാകുന്ന റിസൾട്ടൻ കളർ കുറച്ച് വ്യത്യാസം ഉണ്ടാവില്ല. Okay, so similarly you will be having genetic information from your father in one chromosome and you will be having genetic information from your mother in other chromosome. When these are combining, slight difference will be there. Maybe father നല്ല tall ആയിരിക്കാം, mother may be little short ആയിരിക്കാം, and you will be in between like that. So this is about it. So അങ്ങനെയുള്ള the entire 46 chromosomes which are present in a single cell, that is known as genome. നമ്മുടെ in total ഒരു ഹ്യൂമൻ്റെ ബോഡിയിലുള്ള entire set of genetic information is known as genome. Now what happens when a baby is born? During the sexual fertilization, when the sperm cells or the male sex cells from the father is fusing with the sex cells of the mother, that is egg, that is when sperms and egg fuse and fertilization happens, the first baby cell is formed, zygote. Okay, you can also call it embryo. Okay, for the time being you interchangeably call zygote or embryo. So the first cell, the embryo or zygote is created. Okay, from the first cell, this is the kind of the shape of the cell at the 13 days of growth, at the 28th day of growth. Okay, so from being a single cell, it is continuously developing. Okay, 28 day, 32 day, 33 day, 37 day, 41 day, 44 day, like that is okay, 47 day, 50 day. So the baby is slowly growing. How this happens? It was actually a single cell. the nucleus, the sex cells of the father or the sperm cell got fused with the egg, and the first single cell, the embryo or zygote is formed. Consider it is this one. Slowly that particular cell will enlarge and it will slowly start dividing. One cell become 2, 2 cell become 4, 4 becomes 8, like that it is dividing and a mass of cells is created. And that's why we are enlarging in our body size. Like from one cell, it became a billions and billions of cells. So finally a baby is created in this shape. Now you understand that from the first cell, you got 23 chromosomes from the father side and 23 chromosomes from the mother side. Now when the cell divides, now when one cell is becoming 2, this is the process. You need not study any of the steps in this process. Just for the sake of understanding, for the sake of explanation, I am telling this. No need to study any of the explanation present in these slides. This is actually something related to very depth of biology, mitosis, meiosis, etc. You need not study any of the information. Just understand that through a process of cell division, it includes a number of intermediate stages. Finally, one cell becomes twice. Now, when one cell is becoming 2, the entire 46 chromosome got duplicated into 2 cells. Now, these 2 cells become 4, 4 becomes 8. Finally, it will create a mass of or bunch of cells and a baby is created. Now, since the first cell got divided n number of times and these many cells are created for a baby, the entire cell is having same information. Okay, it's just like photocopying. Right, you got the point, right? So the same genetic information got duplicated and reduplicated and reduplicated, and all cells are having the same 46 chromosomes which are present in the first cell. So this is about the cell division and how a baby is having every part of the body the same set of genetic information. Now the same thing, but I just want to give more clarity. The information in DNA is stored as a code. This code is known as genetic code. That is made up of 4 chemical bases or nucleotide bases: adenine, guanine, cytosine and thymine. So this is the chromosome. If I am taking one particular segment of chromosome, I can see these chemicals arranged in a definite order. See, this is the order, this is the bridge, okay. Adenine, thymine, thymine, adenine, cytosine, guanine, like that. Now just know one more information. Adenine always pairs with thymine, okay, and cytosine always pairs with guanine. This is the order, A, T, C, G. You can remember like that. Now, similarly, we are having about 3 billion base pairs. Then why we all are different? We told that the entire humanity is made up of these 4 chemicals. Our the same genetic information is present, same chemicals are present, A, T, C, G is present in all human beings. About 99.9% of the order is same. If you are opening one cell of mine and one cell of yours, you can see that 99.9% of the order is same. A, T, C, G, C, G, A, T, like that, same. But the 0.1% difference, that makes all the difference. It is said that around, if you are taking 1000 such base pairs, this is 1 base pair, this is 2, this is 3, this is 4, this is 5, like that. If you are taking 1000 base pair, one pair will be different. Okay, the entire set of 1000, okay, up to 1000, this may be same, but the 1001 or the 999, that will be having, in place of CG in my case, you will be having 80. But every 1000 pairs, if it is having one difference, I mean one order different, we are having 3 billion such base pairs. So in every 1000 base pairs of this 3 billion, there is a difference. 1 or 2 order will be different. So that is the reason why we are all different. We are having different shape, different abilities, different kinds of traits and all. And we will come to know in the upcoming slides that if you are getting a dead body from one particular site, now you want to identify who committed that crime. If that is a murder, like in forensics, okay, we see in movies and all, how the sample of the criminal which we obtained from that particular crime site, maybe a blood sample, okay, maybe a blood sample, and the police is having the DNA information of all the criminals, like who have committed previous crimes, okay. Whenever a person is getting arrested by the police, they will be collecting the blood samples and DNA information from that particular criminal's body, and they will be having a database maintained for that. Now, whenever there is a murder, the first thing that police do is that if you are getting one particular blood sample of the accused or the criminal from that particular site, you will bring that to the lab and you will open one cell and you will see the genetic information, okay, the order. Now you will upload that to the database of the police. Like, it's like control of you are doing, okay. You will just copy this particular new information that you got from the crime site and you will search in the database if it is matching with any of the criminal, okay. If the order of the ATCG pairs is matching with any of the criminals in the database, you can identify that this is the person who has committed the crime. This is simple. This is forensics. That's why they are taking these fingerprints or this kind of blood or nail or any kind of bodily information that you are getting from the crime site. You will bring that sample. The forensic people will take that to the laboratory. They will open it up. They will do the genome sequencing. We'll see what is genome sequencing. Genome sequencing is nothing but identifying the order of base pairs. If it is ATCG or ATAT, like that they'll see. And thus it helps in forensics to identify the criminal, to identify the dead bodies, to identify the missing person, to do the paternity testing. Who is the father and mother, like that. Same thing, same basic concept. You will just see the order of base pairs. We'll see that in detail. So that 1% different. So we'll see, we know which part of the chromosome is showing this difference. As I told you, you are having 46 chromosome. Now the forensic people know that the 21st chromosome, in the 21st chromosome, there is a segment of DNA. In every people, that particular area is different. Like if the forensic people know, if out of the 46 chromosome, in the 21st or 22nd chromosome, if I am zooming in, if I am taking that particular gene, if this area is different in every people, okay. Now I got a blood sample from the crime site. I am searching that in the database. Only let this be the person's, let this be the gene that is present in the blood sample that I got from the crime site. Now 3 people are suspected. Now I am uploading this particular information in the police database, but I found that in all other people this area is not matching. That means 3 people are, like all the people are different. But in the database, if I am getting only one person in this particular area, his is also same, A. In every person, this particular area must be different. But if the person is same, the same will be A only here, okay. That will be showing, matching. That's why in that method we can identify this particular person, both are same. His sample we got from the crime site. So this is the thing. So this is the importance of order of this genetic basis. We can identify a person by just having 1 or 2 cells from the site of this particular incident. Maybe a disaster site, like a building got collapsed and like many people died. Now when we try to identify that particular person, he is not in an identifiable position. Maybe he might have got burnt or something like that. Now his parents want to see that whether this person is their son. Now what we'll do, we'll take the cells from parents. Now we will match that with the person, the dead body we got from the site. Now we'll do the paternity testing. Okay, if maximum genes are matching, that means this is his son only, like that. So this is the importance of this order of genetic pairs. The same thing, but explained in a different manner. You can see that because of 1 or 2 letters of like difference in this particular order, 1 or 2 difference in the ATCG pairs, some people's eye will be blue, some people's will be light brown, some people's will be dark brown, some people will be some kind of light greenish. So this is all because of 1 or 2 letters change in the order of chemical. Got the point, right? Now genome sequencing. Genome sequencing is the same thing in which we are determining the order of the nucleotides. we will open up a cell in the laboratory setup. We will identify the order of this adenine, threonine, like cytosine, guanine pairs, and this order will be different in different person. So if I am having ATCG, in your case it will be AT or CG like that. Okay. So this particular observation or this particular identification of the order of the base pairs, that particular technique is known as genome sequencing. Right.